on to our last talk, which is Arwen Gao. Who will tell us something about the lysosomes.
Good afternoon, everyone. Hope you are still awake. It's almost the end of this session. So first of all, I would like to thank the organizers giving me the opportunity to present my work here. My name is Arwen, and today I would like to talk one of the projects I have when I was a postdoc at Johan's lab in the EPFL in Switzerland. So this is about a lysosomal surveillance response, in short LySR, that reduces proteotoxicity and extends the health span. So as you all know that stress response are tightly linked with aging. And among all those different organelle-specific stress responses, it seems like none of them takes the lysosome as the center and directly check its effect to directly effect on the aging. So most of the people that in the aging field are only checking lysosome changes because they wanted to see if there's anything related to autophagy, mitophagy, or lipophagy, and also of course mTOR part. So we question ourselves whether there's a direct link if we target lysosome that can also affect the aging process. So we know that many substrates are degraded in the lysosome by many different enzymes, and those enzymes require an acidic environment with a low pH. And this pH is carefully controlled by VADPase, which is a proton transport that mediates the hydrogen into the lysosome to maintain the pH. And in the worms, there are this VADPase is assembled by the two domains, which is encoded by 20 genes for the different subunits. And we want, because of the importance of the VADPase for the lysosome function, we wanted to first to knock down each of these subunits in the worms and to see what are their effects on the aging process. So interestingly, what we see is that knocking down some specific VADPase subunit genes, including VHA6, 8, 14, 15, and 20, they extend lifespan by about 60%. Whereas when you knock down VHA16 and 19, that shortens lifespan. To understand where the difference can explain the long-lived and short-lived phenotype, we perform the RNA-seq analysis, and what you can see from the PCA plot that the controls are separated nicely from the short-lived and also the long-lived ones. To focus on the ones that can explain the difference we see in the long-lived phenotype, we focus on these 760 genes that are exclusively upregulated in the VHA6 RNAi worms. So what do they change? So you can see that most of the genes that are upregulated in the VHA6 RNAi worms are mainly involved in the lysosomes, proteolysis, metabolic pathways, and innate immune response. Many of them are actually homologs for mammalian cathepsin proteases, such as the CTSB, A, G, and E, and also some other ones that are also the proteases. And you can see that many of these VADPase genes are actually upregulated in the VHA6 RNAi worms. But you can also appreciate the difference compared to the long-lived ones. The short-lived VHA16 and 19 RNAi worms, they have an upregulation of the autophagy genes, meaning that they really have different metabolic profiles compared to the long-lived ones. So now we wanted to introduce you this new longevity pathway that we call it lysosomal surveillance response, in short LySR, which is a unique longevity-linked transcriptional response. It's typified by a strong induction of many genes involved in proteolysis and the lysosomes. And then we wanted to discover if this new long-lived pathways if any of the canonical longevity pathways can explain the difference we see in this long-lived phenotype. And clearly, when we try to knock down VHA6 in all those mutants that are models for insulin signaling, AMPK pathways, mTOR pathways, the E2 mutant, which is a caloric restriction model, and also the mitochondrial stress response, you can see that the RNAi of VHA6 clearly extend the lifespan of all those mutants, suggesting that our new LySR activation is independent of those pathways. And to visualize this LySR activation, we noticed that we could use this CPR5 GFP worms, which you can monitor the intensity of the GFP. When you knock down VHA6 in these worms, you can clearly see a big induction of the CPR5, and this is totally different from the short-lived ones. So we can use this to monitor and also screen for regulators and activators of LySR. Because of the time limit, so I will just keep this part short. Because after we have this activation of LySR, we wanted to study which are the transcription factors are important to govern this pathway. So we did a screening, and now we found that the GATA transcription factor ELT2 is the main regulator for LySR activation. And we also validated by the lifespan, seeing that after knocking down ELT2 in the VHA6 RNA worms, it's abolished the lifespan extension we see in the VHA6 alone. And next, because we see that a lot of proteolysis genes are upregulated in the LySR activated worms, that means that we probably can also try to use this mechanism to see that can reduce the protein aggregates in some disease models. So the first one we tried is the Alzheimer's disease worm, which is a GMC strain in our hand. You can clearly see that the VHA6 RNA really reduced the beta amyloid aggregates in the GFC worms, which is almost similar to what we see in the CL control, which is not expressing any beta amyloid deposit. And this is also dependent on the ELT2, which you can see that after ELT2 is knocked down, you can see this clearance effect is also abolished. And afterwards, we also measure some physiological related phenotypes, for instance, the worm mobility and also the paralysis of those worms. You can see that VHA6 is promoting their movement and also reduce the paralysis. And this is also in an ELT2 dependent manner. So after that, we extended our investigation to a bit more to the mammalian direction. And what we did is that we used this SY55 neuroblastoma cells that overexpress the Swedish APP protein, which is the amyloid precursor protein. And you can see that overexpressing the GATA transcription factor 4 and 6, which are the homologs of worm ELT2, clearly reduce the beta amyloid proteotoxicity in the cells. And this is also coupled with the upregulation of those catheptic genes, which are the proteases to clear up the protein aggregates upon the overexpression of GATA4 and 6. To summarize this, what we found is a novel lysosome surveillance response, which after activation in the worms, we can see that the ELT2 transcription factor is the main driver for this pathway. And upon LySR activation in the worms, a lot of genes involved in the lysosomes and proteolysis, such as CPR5 or 8, that they are upregulated, and then that can promote the proteostasis and reduce the protein aggregates, which can also lead to extend lifespan in the normal worms and also promote the health span of the GMC worms, which is the Alzheimer's disease. And in the mammalian part, we see that overexpressing the ELT2 homolog, GATA transcription factor 4 or 6, can also give us the similar benefit effect. And you can see that the lysosome-related genes like cathepsin gene, CTSB or D are also activated, and that promotes the clearance of toxic aggregates. So I would like to thank my supervisor, Johan Alwerks, for his great support and supervision, and also the other main contributor for this project, Terry Lee, and our other colleagues in the lab that helps us to finish the projects, and also our collaborator from BCM, Rachel Airy, who helped us to also confirm the VHA6 phenotype in her Alzheimer's worm model, and our collaborator from Nagi Bioscience that testing our LySR activation on their worm-on-chip techniques. And I would like to also advertise some posters. So the number 31 is from Gabby, who used unbiased worm population study on the aging and also UPRMT study. So if you're interested in genetic studies on lifespan, please come to visit that. And then there's also 34 from Man, who's also studying the novel mechanism that explain the longevity of the MRPS5 RNAi, and another 2 compounds that are also novel, and one of them it will be given by Rebecca, who's giving also a talk tomorrow, so please save your time for that. And also mine on number 13, so thank you very much for your attention.
Thank you so much, Arwen. Great talk. Maybe I can start with a question. So how big an overlap is there between LySR and the UPR?
Actually,
is there an overlap?
There actually is, because we found LySR first because we studied mitochondrial stress response, the UPRMT. And after that, we wanted to understand how the paralysis is regulated, and we found this is also regulating the UPRMT activating worms. And then we realized this could also be upregulated independent from that. So it could be directly targeted or activated by targeting the VHA6 gene, or it could be like a downstream of this UPRMT pathway. Okay. So that was the case.
All right, very cool.
Yeah.
Other question for Arwen? Otherwise, you can post questions on Slack, and I just want to give a round of applause to the amazing speakers we had in this session. Please go and speak with them during the break. We have a poster session now. Please look at the posters, and there are also posters upstairs. So if you walk up the stairs, you will not only get smarter when you get to the top, you're going to get in better shape and become younger.